All right, folks, let's go ahead and begin. Really excited to have you joining us today. My name is Emily Taylor. I'm one of the organizers of the first ever World Snake Week. There are three of us who got together to organize this, reasoning that World Snake Day, which is today, Happy World Snake Day, was not enough. We needed a whole week during this year of the snake. And those three of us are me representing Project Rattlecam, the first ever live streaming camera system on rattlesnakes, Melissa Amarello from Advocates for Snake Preservation, and Christina Zdenek from the Australian Reptile Academy and also the IUCN Snake Specialist Group. We got together and brainstormed some ideas for getting groups, snake positive groups from around the world involved in sharing and amplifying all of our presentations, outreach events, webinars, and more all throughout this wonderful week from July 13th through 19th. We hope this will continue in the future, and we hope everyone will stay involved. So for today's webinar, please keep your mic muted throughout. And here is some information for you. This is the second of 6 official webinars that we have scheduled for you for World Snake Week. We had Ram Whitaker on Monday talking about cobras and his life in India. Today we have Dr. Rulon Clark that's coming up. And shortly we're going to have Dr. Rick Shine at 5 p.m. Pacific. Tomorrow we have Dr. Jesus Rivas and Jazz Ong. And then on Friday morning we have Hiral Naik. In addition to all of these, there's another set of 7 talks, shorter talks, that was organized by the IUCN Snake Specialist Group. All 13 of these talks are available for pre-registration on snakeweek.org/webinars. So as you're watching this presentation today, I want to urge you to all keep your mics muted the whole time and to enter any questions that you have for Dr. Clark in the chat. Feel free to enter questions as they come up. He's not going to answer them as they come up. We're going to kind of wait until the end, and then I will do a moderating of the questions at the end. We're going to finish up within the hour. We'll finish up by 4 p.m. Pacific Standard Time. This presentation will also be posted to YouTube in the coming days on our Snake Week YouTube channel. So I'm really excited today to introduce Dr. Rulon Clark. The way we worked this out was Melissa, Christina, and I, the organizers, all got together and brainstormed, and we said, who do we most want to hear talk about snakes? Who are our favorite speakers? And we divvied it up so that we tried to get someone from every continent. We actually ended up getting 2 people from Asia and no one from Europe, but that's another story entirely. That will change in the future years. But for now, I want to share that I got to pick North America, and I picked Dr. Rulon Clark. It's my favorite presentations of all of North America. And so he's here today to talk to us. Rulon got his bachelor's degree at Utah State University before going to Cornell University, where he studied timber rattlesnakes and started making big waves on the venomous snake scene. And I met him. We were both grad students at the same time, but I was across the country at Arizona State University. And we've been friends and colleagues ever since. And so with that, Rulon, you can go ahead and pull up your slides now.
Yep. Let me, let me share my other screen. Okay, so all right, Emily, are you seeing my screen with the slides now?
Yep, it looks perfect. Take it away.
Okay, great. Thank you, Emily, so much. I'm really honored to be here, and that's such nice things you said about me. I know it's been actually amazing, our friendship, because like Emily said, we've been kind of paralleling each other ever since grad school, and then pretty excited that we both ended up in California. Actually, we're close enough that we cross-fertilize students all the time. We see each other a lot. We go to a lot of the same meetings. It's been a really great friendship. I'm really excited to be here for Snake Day and for Snake Week. And I thought that what I would do is sorry, my screen was glitching out there for a second. Emily, you're still seeing me all right? Okay. Um, so I thought that what I would do is kind of talk about, and Emily, if I do freeze or anything, since you're the only one who can unmute themselves, just let me know if something's going wrong with my audio or anything like that.
I'll speak up if anything's wrong. You're good right now.
Okay, cool. Um, so I wanted to talk about some of the projects that my lab is doing that are focused on snakes, and particularly on kind of observation and natural history of snakes. Um, whenever I can get away with it, I try to work the term natural history into a talk title because it's kind of a core part of my identity as a scientist. And I think that term means kind of different things to different people. But at the core of it, I think is kind of detailed and careful observation of organisms in nature, which is pretty much my favorite pastime. I mean, no offense to everybody here, but I'd rather be watching snakes than talking to you about watching snakes. But this is a good close second. I also think of natural history as being quantitative, but not necessarily hypothesis testing. It's more free from, it's like quantitative, but free from the constraints of testing specific hypotheses. Um, it's kind of, you're allowed to just wonder and record and observe and document, but you're trying to drop preconceived notions about what might be going on. And I think this philosophy, I inherit this a bit from my academic mentors. I was trained by some really influential and prominent herpetologists, including Harry Green, who has been a really strong advocate for snakes and natural history. And he was actually trained, his doctoral mentor was Gordon Burkhardt. And if you haven't seen Gordon's book, here's a picture from back in the day of Gordon and Harry. If you haven't seen Gordon's recent book, The Secret Social Lives of Reptiles, with Sean Duty and Vladimir Dinets, excellent book, highly recommend it. They have a section in there where they talk about the importance of kind of quantitative natural history studies, and they have a really detailed and compelling argument that focuses on the problem with this overemphasis on experimentation in a controlled laboratory setting, and this increasingly narrow focus in science on hypothesis testing and pre-registering experiments, and not putting enough resources towards just observation and natural history and these observing things free from those constraints. I think they make a pretty good case that this isn't just specific to, like, snakes and herpetology. This is true of any biological system where we don't know enough about how it operates naturally to make informed hypotheses. And unfortunately, a lot of reptiles are like that. We just don't know enough about how they operate naturally in the free-ranging world to really necessarily make informed hypotheses yet. So anyway, I'll come back to this at the end of my talk, this idea about quantitative natural history or next generation natural history. I'm actually optimistic about kind of the growth of this area in the future. But for now, I'll just emphasize that it's, for my own personal kind of perspective and my own development as a scientist and the questions I ask, when I look back at why I got into certain things and where that came from, it almost always comes back to observations of animals in nature. As kind of the first example here, I got into herpetology as an undergraduate. So I did my undergraduate degree at Utah State University. And another very famous herpetologist, very influential, great guy, Dr. Ed Brody, the older Ed Brody, was my advisor there. And I worked with him and some of his grad students on a project in Arizona where we were looking at flat-tailed horn lizards. And we were also like observing and recording data from like other animals. And one of the ones that really fascinated me was sidewinder rattlesnakes. I didn't know it at the time, but Brody and some of his students kind of showed me that you can follow the trackways of these sidewinders. They have these really specific kind of tracks that they leave in the sand due to their unique like sidewinding motion. So you can kind of tell where the animal is going and follow it. and I mean, what a great snake. That just seemed magical to me. And I would spend a lot of my free time just combing through like the sandy parts of the desert out there, tracking sidewinders so I could find them and just look at them. I wasn't studying pit vipers. I didn't have like equipment to capture them or anything. So the best I could do is just kind of watch them when I did find them. They were almost always doing this. You can see this snake here is just coiled and pancaked kind of into the sand, hiding often like under the edge of a bush or outside the burrow of a rodent. And it seemed pretty clear to me, even with my limited information at the time, oh, that's like a strategy. Must be a hunting strategy. They're just waiting there like a predator in ambush, waiting to just leap out and attack a lizard or a small mammal if it comes by and gets close enough. And so that interest, those observations, when I went to do my own graduate work at Cornell, my advisors there were, Harry and Craig Adler were and Tom Eisner to some extent. They were all really open about the importance of natural history and observing animals. And they were also just like, just do what you want to, they didn't have any like preconceived notions about what I should be studying. They're just like, study something that interests you. And after kind of, casting around and looking at ideas, I came back to these observations had made about sidewinders. And I decided what I really wanted to do was try to study in detail that process of ambush hunting and pit vipers and venomous snakes. I thought it would be really, there were some interesting things about it that I could discover. Working more with a snake that was local to upstate New York, where I was, so the timber rattlesnake. And those observations and the studies that kind of stemmed from them, that shaped my academic career ever since. So I've been kind of, continually spitting out projects and studying things about this hunting behavior of pit vipers. So let me talk a little bit of details about that, then I'll go into a few more specific studies. So this kind of shows the cycle, what I think of as the kind of the hunting cycle for most pit vipers that are ambush hunters. And I'm gonna, I'll show this with sidewinder rattlesnakes since they were kind of influential in getting me on this path. So here's a sidewinder that is moving out at the beginning of the night, starting this process of an active search of the environment. And when a snake is hunting, it's probably looking for cues to lead it to an ambush site. So it's probably, using its chemosensory abilities, its tongue flicking to try to find somewhere where it can sit and wait for prey to come close enough. And when it does, it then like buries itself in the sand. Like here you can see the sidewinder with its eyes just up right there. And then it sits and it waits. And this is the part of the snakiness that really kind of dominates, is really alien to us, is the amount of time that they can just sit still waiting. Is kind of a superpower for them. They can be so patient and so cryptic. Here's one of our field videos where I'm recording an ambushing snake. You can see the snake right here. You can see the time clicking by here. This isn't fast forward now. And you can see the snake is just sitting there like stock still as the morning progresses. He's still kind of just up there coiled. This is actually a Mojave rattlesnake rather than a sidewinder, but he's hidden in his outside this burrow in this little patch of grass. And the sun, the lighting is starting to change. You can see that the sun is coming up. Every now and again, there are small like insects or other invertebrates that buzz by, and the snake just waits through it all patiently. And you might do this for several nights on end, many nights in a row without really anything happening, without having an encounter until he finally, if he's positioned himself well and he's lucky enough, some animal will come close enough that he can jump out and strike at it and try to eat it. So very long, extended period of waiting. But if something does come close enough, then the snake is capable of going from nothing to very, very sudden movement very quickly. So here's a high-speed video slowed way down so you can see the strike of a sidewinder as it moves towards a kangaroo rat. This all happens in about a quarter second. But if I slow it down, you can kind of see that the snake, as it moves forward, it rotates the maxillary bone and pushes those two fangs forward so that it can puncture the body cavity of whatever it's trying to bite. If it's lucky, it lands those fangs in the animal, and here's the approaching head of the snake towards the kangaroo rat, and it'll squeeze venom into the animal during the amount of maybe half second or so that it's injecting, and then it will release typically and let the venom take effect. So most small animals like kangaroo rats, most small mammals have pretty sharp teeth and claws and will readily defend themselves. So the snake doesn't want to like hang on to that thing as it dies. It wants to let it go. And then the animal runs off and dies somewhere. And the snake undergoes this process of strike-induced chemosensory searching to find the carcass. This is kind of a specific type of chemical searching where the snake is using both cues from its own venom and cues from the scent of the animal itself that it bit to locate the scent trail, that specific individual, and it can follow a dying mammal for many meters until it finds the carcass. And that process can take hours, maybe sometimes even days. The snakes are very persistent in following that scent trail because they want to find that carcass. It's they don't eat very often, so any given opportunity is a huge opportunity for them. And then, of course, snakes, rattlesnakes, and other snakes are famous for being able to ingest relatively huge prey items. So here you can see the Sidewinder has successfully found a kangaroo rat carcass. He's beginning the process of ingestion, walking his head, kind of using those mobile bones in his mouth, or the roof of his mouth and in his jaws, to kind of walk his head over the carcass and then pull the carcass down into his gullet while he'll digest it. And that digestion phase can actually last several days to weeks, depending on how big the prey item is. And then at the end of all that, after they've digested the meal, the snake will become active again, and the kind of restarts the process of, once they're hungry, go out, look for cues, try to ambush something. So I have research projects that kind of stem from all different aspects of that process. I'm gonna kind of pick and choose a few that are of current interest that are ongoing, and just kind of talk about some of the details. And I'll start with one of these that has to do with kind of this active search for prey cues or waiting in ambush. I mentioned before the snakes are probably using vomerolfaction or olfaction, the tongue vomeronasal system, or their sense of smell. They're also very tuned into visual cues, and with pit vipers, we have to say those are thermovisual cues, of course, because one of the things that makes them so fascinated is they have this unique and interesting thermal imaging system, this pit organ system. I got into this again because of observations or experiments we were doing with Northern Pacific rattlesnakes in Northern California. I was working with Bree Putman and my students Bree and Matt Barber at the time, looking at interactions between rattlesnakes and ground squirrels, the California ground squirrels, looking at kind of predator-prey signaling and predation behavior. And there was a paper that came out at this time where one of Don Owing's students, who was a colleague at that time, pointed out that ground squirrels that are signaling towards rattlesnakes with their tail, they have a tail flagging behavior that they do, often had like a warmer tail than if the squirrel was not signaling towards a rattlesnake. And then you could, maybe that indicates that it's sending like a thermovisual cue, not just a visual cue, but maybe there's a thermal aspect towards the predator that is one of the few animals that could actually see that body heat. And of course, we can see that. So this is an example of us using technology to be able to look a little closer and deeper. This is a visual of a squirrel body and tail being taken with a thermal imaging camera. Here's another thermal imaging camera of a squirrel. So I'm recording a squirrel with a thermal imaging camera and a regular camera side by side here, same squirrel. And the thought in the people who study snake sensory systems is that while the snake is seeing a composite of these two things. Its pit organ allows it to detect thermal contrast, just like the lens of the thermal imaging camera. And then that's integrated with its visual system and the optic tectum of the brain, since both sensory systems talk to specialized brain nuclei that receive input from infrared and visual systems. So it's thought that this infrared organ forms kind of an integral part of the visual system, thus giving snakes like that same, maybe the same type of view that a thermal imaging camera might have. But we were experimenting at the time with some ground squirrel models and tail temperature and body temperature, and it wasn't going very well. The snakes it seemed to kind of ignore temperature variation in the experimental models we had, at least, and it was frustrating. And I started to wonder more about, like, what is it like to be a snake that has this thermal imaging ability? Like, what are they really able to see with it? And I remember having a very important conversation with my colleague George Bakken, who had spent decades studying kind of the pit organ system. And as he has pointed out in some of his papers prior to all this, when I talked to him, went back and looked at some of his papers, yes, the pit organ system maybe is a part of a visual system, but it is a really poor camera. It's like a pinhole camera that doesn't have a lot of depth to it, and doesn't have a lens mechanism, and doesn't appear to have any way to actually generate like a highly resolved image. So George did some biophysical modeling. This is a thermal image of like a mouse or a bird on a log at night in a forest with a thermal imaging camera, where the animal is kind of nicely resolved against the background. And these are some models that George made of what the pit organ might actually see, on making some different types of assumptions. But even with generous kind of assumptions, it's really imprecise. It's a very, very blurry image, if you want to even call it an image. Of course, a biophysical model like this doesn't actually tell us what the animal is seeing necessarily, because we don't know what type of, how that information might be processed in the central nervous system of the animal. What the inputs might be different than what the snake perceives, based on there are various hypotheses for how a snake might process crude inputs like this to sharpen the imagery. I was also working at the time with a student, Hannes Schraft, who is very interested in these types of questions of understanding what the umwelt of a snake with a thermal visual system might be like, a snake with a pit organ, how it might be able to perceive the world. And Hannes come up with a very clever experiment we could do, again, with sidewinder rattlesnakes, where he just simply asked the question, like, well, if the pit organs are part of the eyes, if you take away the eyes, can the pit organs kind of act like eyes and allow the snake to complete a very, very simple visual navigation task? Turns out this works pretty well. If you take a sidewinder and put it in the middle of a big arena that we had fenced off, it wants to hide under a bush, and it can see those bushes even at night if it has its eyes. But those bushes also are strong. They stand out for their thermal qualities as well as their visual qualities. They have a lot of thermal contrast with the night sky, and so you should be able to see them pretty well with just the pit organ as well, even if it's a blurry image. Like, this is the George Bakken view of what the snake would probably see versus the thermal imaging camera. And so we did this experiment. We essentially put a lot of snakes through this simple trial to see how quickly they could navigate to the bush, and versus when we didn't cover their eyes or their pits, versus when we did cover both eyes and pits. Maybe not surprisingly, if you cover their eyes and pits, they have trouble. They don't move very straight. They wander around, and they have trouble finding the bush, compared to when the eyes and pits aren't covered. But very interestingly, if you do cover their eyes, they do just as poorly as if you cover both eyes and pits. That is, covering the eyes means that they can't effectively kind of navigate this task, even though they have the pits available. if you cover the pits, it doesn't really affect them. If they have the eyes available, then they do the task just as quickly as if they didn't have the pits available. So in this context, at least, it was pretty clear that they're not using the pits the same way they're using their eyes. Their pits aren't substituting to allow them to compensate and complete this task. So that was, one experiment. Hannes also did this experiment with a thermal imaging camera looking at a different mammal species. We wanted to see, kind of like with the ground squirrels that became aware of a snake, if we just looked at a different species, one that isn't signaling with its tail, but a kangaroo rat that becomes aware of a snake. If we experimentally expose a free-ranging kangaroo rat to a rattlesnake predator, it becomes aware of it, and its body temperature does change in various ways. It's a pretty subtle change. You have to, not a drastic one, but you can see it and measure it with a thermal imaging camera. But we didn't really have any idea whether or not that meant anything to the snake. The snakes, we still were left wondering, well, do the snakes have the ability to resolve these kind of subtle temperature changes, or is it just that that's not what the system is designed for? So that led us to kind of go back into the lab. We were doing a bunch of stuff in the field, but we wanted to complement that with some more focused kind of laboratory studies. So we wanted to go back in the lab and kind of ask a very specific directed question, which is, how well can a snake resolve a moving thermal target if it doesn't have any visual cues, if it doesn't have any other information about the target other than temperature contrast? So George Bakken visited our lab, and along with Hannes and myself and some other students, we put together essentially a snake vision testing device that would allow us to ask a snake that is in a sensory deprivation chamber, can you see this image, this thermal moving thing? The way it works is there's a snake in a box, and then we would put it in a room, would turn off all the lights, and it was an isolated room. And there was a background plate that we can control the temperature of remotely through the water bath. And there was a pendulum that we could release from an electromagnet that we also controlled remotely. And that pendulum, we could remotely adjust the temperature so that it was different from the background. So it gave us the ability to present a silent but mobile stimulus to a snake that was just sitting there staring at a thermal background. So what this looks like here, I've filmed the background plate with a thermal imaging camera here, so you can see the release of the pendulum. And you can see that the thermal camera can very clearly see the temperature contrast there, that the temperature, that pendulum was about 6 degrees warmer than the background. And then we would use a camera, a night vision camera that was watching the snake, to record the response of the snake as we exposed it to those different pendulums. So here's a snake in the box. I'm gonna play this. Hold on, I think it's playing now. There you can see, if you watch carefully, you can see the snake flick its tongue a few times. Let me see if I can play that again. So when you see the pendulum come down in front of the camera, the snake kind of moves its head and flicks his tongue a little bit, indicating that's a positive response, versus when the pendulum is the same color as the background or the same contrast as the background, then often the snakes can't see it and they don't respond. They just kind of sit there. So we were taking advantage of this like evoked behavior on the part of a snake that is sitting there in the dark looking at a background. It turns out if anything interesting comes up, it doesn't have to be a scent, anything that comes into its visual or thermal field, it'll do what snakes do, which was flick your tongue at it, see what it is. And so we showed with this temperature contrast that we could very clearly get very strong responses from snakes that were looking at temperatures that were warmer or cooler than the background. So any sort of thermal contrast that was moving, strong responses from the snakes, and no responses if the thermal contrast started going to around 0. And more thermal contrast in general up to a point stronger response from the part of the snake. So we had a pretty good sense from this that, hey, this is working. We can use this to ask the snake, can you see this? And so then we made it more challenging by putting a metal plate in front of the background, so the background became a gridded background, so that the snake would have to be able to resolve the spatial grid in the background to see the pendulum. I'll show you what I mean by this. The principle is the same as your visual system. Like if you can resolve the black and white bars here, you see them as distinct. But as they get further and further away from you, at some point you're not going to be able to resolve that those are actually separate black and white bars, and you see it as the average hue of black and white. So you'll see it as like a gray color. So we did that with our background, and I can show that by showing a thermal image of the background. This is with the thermal imaging camera where the camera is defocused. So at the beginning of this, we've unfocused the camera, so the camera isn't actually resolving the gridded background right now. But if we focus the camera, you can see the background is gridded, and now the camera also resolves the fact that there's a moving pendulum in front of it. Whereas if the camera can't resolve the background, that moving pendulum, it's set to be roughly the average temperature of the hot and the cold spots in the background, which is why it disappears when the camera doesn't resolve the background. So that gave us the ability to then look at snakes and ask them, what do you see when we make the grid coarser and coarser? The visual system should be able to resolve a very coarse grid and tell that the pendulum is actually moving in front of it. And we can do this with like different levels of thermal contrast. And so you can see here's the results from when there's no grid. It's just like a homogeneous cool background. As long as the pendulum is slightly warmer to much warmer than the background, the snakes respond really strongly to it with that tongue flicking response. So then here are the different grid sizes. So I can show this is a result of like 300 or 400 separate experiments with a bunch of different individuals to show that this happens, which is what it looks like, that the snakes don't respond to pendulums in front of gridded backgrounds when the pendulum is at the average temperature of the warm and cool spots. Even with very, very coarse grids, they're still not resolving the details enough to be able to have a statistically significant response, a response that's different from zero, from no contrast between the pendulum and the background. So all of this, I was worried that the snakes had habituated, so we actually retested them at the end of all of this. But if we took the grid away, they're very responsive again. So the post-test indicated that it's not that our snakes are like habituating over these repeated tests that we're doing with them. They're still will respond if they can see it. I think it's just that they can't detect it, that their thermal system, like George originally had kind of hypothesized, it does a poor job of resolving background details. And so they really need, this kinds of emphasizes to us that the snakes really need to be looking at kind of a homogeneous, cool background in nature if their thermal imaging system is going to help them resolve something important that might be moving around there. Or maybe we're kind of wrong about it being integral to the visual system. Maybe it's more like an alarm system, like a burglar detection system. It is very sensitive to motion, so maybe it's just any type of interference that can detect kind of wakes the snake up and makes it focus on something. At any rate, there's it's kind of confusing. But that's interesting to me. It's interesting to kind of think about, well, if this isn't a very good like visual spatial resolution system, what do all these snakes use it for? Because now if we kind of go out beyond rattlesnakes and start thinking about boas and pythons, there's several lineages of boas and pythons that have all sorts of diversity in infrared sensing pits. So the pit vipers, the boas, and the pythons all kind of independently evolved versions of this thermal imaging system. And there's, the number of pits, the placement of pits, the type of pits. There's a crazy amount of diversity that we don't really understand what it's for in the boa and python lineages. So I'm working with current students to try to tackle some of that, hopefully. Okay, so kind of moving on from that, there's another vignette I wanted to get here about this. That again stemmed from observations we were making in nature about snake predatory behavior. And this had to do with the predatory strike. And this came from our kangaroo rat studies. We're also studying kind of predator-prey interactions between kangaroo rats and snakes. And this was with sidewinder rattlesnakes and kangaroo rats in the Mojave and Sonoran desert regions. And we kept seeing kangaroo rats we were recording this with like regular cameras and using the kind of our unmanned field observation equipment to make these recordings. And we kept seeing kangaroo rats survive bites. If you watch carefully, there's a sidewinder that with the regular camera, it looks like it bites a kangaroo rat. That's essentially what a successful predatory strike from a snake looks like. It's very fast. They release the animal, but you can see this kangaroo rat is like unaffected. It's like just running away. And we were wondering, like, are they resistant to venom? Like, or we actually had a colleague, Matt Holding, did some tests for us and indicated they don't have high levels of physiological resistance to venom like ground squirrels or wood rats. And so we figured, well, they're probably surviving these bites because the snakes aren't actually maybe injecting venom or maybe biting them. But with the regular camera, we just couldn't resolve the details. We're trying to watch these interactions, but it's with a 30 frames per second camera, it's all just like a blur of motion. So our colleague at UC Riverside kind of loaned us some of his high-speed camera equipment, and with Tim's support, and then with two other students, Grace and Malachi, working with me at the time, we decided we're going to take these high-speed cameras out into the field and figure out what's going on by filming some of these predatory encounters with kangaroo rats to see the strike. And this worked. It was a lot of effort. There's like 500 pounds of equipment that had to be hauled out at any given snake to try to film this, but we did end up getting enough data to make some sense of it. We filmed it simultaneously with two cameras, so this video is kind of showing the same kangaroo rat hopping in front of, this is actually a Mojave rattlesnake in this case, in slow-mo because it slowed way down since we recorded it at 500 frames per second. And you can see very clearly that the very brief contact between the snake and the kangaroo rat is brushed off by the kangaroo rat because it's doing this high-powered evasive jump, getting out of the way of the striking snake. So Grace and Malachi published the details of this if you want to check that out, but I'll just briefly kind of summarize some of what we found here. If we break down the outcome of these, we had like 30 odd interactions between snakes and kangaroo rats that were predatory strikes from the snakes that were recorded in high speed so we could actually see the details of what happened. And about 20% of the time, the snake, we probably won't be able to see this with a regular camera, but we could see pretty clearly that the snake kind of mistargeted. It either came up short or it was targeting a space like around the kangaroo rat. It would have missed even if the kangaroo rat hadn't jumped out of the way in some proportion of these. And then maybe 25 to 30% of the time, the kangaroo rat dodges. So the snake would have hit it. So this is an example of a snake that probably would have been able to land its strike, but the kangaroo rat reaction time is so crazy quick that it powers itself out of the way and the snake misses. And that high-powered evasive jump, it sends the kangaroo rats flying through the air like up to a meter and a half. It does come down here over there. That they typically, the snake has kind of blown its chance. The kangaroo rats land and then they just bust out of there. So when the snakes miss, they don't get a meal. And then, interestingly, about half the time the snake does contact the kangaroo rat, but a half of that half, so 25% of the time, the contact doesn't look like it's long enough for the snake to inject venom because the kangaroo rat contorts itself in a way that causes the snake to release it, or it just kicks the snake off. In this case, the kangaroo rat twists its body and uses its hind legs to kick and dislodge the snake. So it sends that snake flying away, and that snake was in contact with the rat for only a few milliseconds, probably not enough time for it to actually effectively inject venom, and that kangaroo rat runs off and survives. So the poor snakes are left with being successful only about 25% of the time, but they're low energy specialists, so that's still enough actually for kangaroo rats to be an important prey item for them, even though kangaroo rats have evolved all these sophisticated detection and evasion strategies as part of their predator-prey interaction. Anyway, that last kind of area, that's where it kind of got us interested in the details of the relationship between kangaroo rats and rattlesnakes, and details of like, well, how much does it matter if the snake is foraging at night versus the day? How warm the body temperature of the snake is? Might that affect the speed of the snake and the ability of it to land a strike before the kangaroo rat avoids it? So we started thinking more about snake kangaroo rat interactions and body temperature. And this led to a project with two other grad students, Ryan Hanscom and Jessica Hill worked on this with me for the past 4 or 5 years, where we're looking at the relationship of one particular pair. So kangaroo rats and rattlesnakes are interesting because they're co-distributed throughout the arid regions of the Western North America, and there's several species pairs that are found together. And we chose to look at the prairie rattlesnake and the Ord's kangaroo rat, because Ord's kangaroo rat eat a lot of, or prairie rattlesnakes eat a lot of Ord's kangaroo rat. So it's a primary prey item in areas where they co-occur. And they co-occur over this really wide latitudinal gradient. So they can be found all the way from like Texas up into Canada. And, so they're found together where the snakes are in very thermally challenging conditions versus where snakes are in more desert, what we might think of as more favorable conditions in terms of snake body temperature. And so this got us into like this project that was looking more broadly at kind of endotherm, ectotherm interactions, using these two animals as an example, thinking about what is climate change going to do to a tight-knit species interaction when one of the members of the pair has a body temperature that is dictated by the environmental temperature, and it's going to be subject to all changes in those temperatures as the climate warms. And kangaroo rats, we also argued that kangaroo rats are important to study just because they're such important keystone ecosystem engineers. In areas where they occur, they build really extensive mounds all throughout the habitat that lots of other animals use. And they're granivorous, like seed dispersers. So, ecologists have known for a while that if you start messing with kangaroo rats, it changes the vegetation community. So we figured this would be important to understand the interactions between snakes and kangaroo rats, because snakes themselves are, even though any individual snake is a low energy specialist, they occur at really, really, really high population densities compared to other types of predators. This is our site in Texas, where there's like 3 large-bodied rattlesnakes that co-occur there, and all of them are fairly abundant. And this is just scratching the surface of the number of animals that are actually there. These are the ones we actually found and know about. And if we use market capture to estimate population size, there's hundreds and hundreds of rattlesnakes in this several kilometer patch of desert that are out there trying to hunt small mammals. Anyway, this project is trying to look at interaction rate and seasonal and diel activity patterns and spatial ecology of snakes and how temperature affects that. And so the last thing I want to talk about is how we're trying to do that. We're using animal-borne accelerometers to try to get some of these very detailed behavioral data from free-ranging snakes. The idea here, other people have been doing this with mammals and other species for a while, You attach a device to the animal, a small acceleration device that records acceleration signatures in 3 dimensions, at least 3 dimensions, like X, Y, and Z. So the sway, heave, and surge. And then you use machine learning models or other AI to decode the acceleration signatures that match like species typical behaviors like running or walking or digging, etc. And our colleague Dominic DeSantis started piloting this work with Western Diamondbacks several years ago. And so we have worked with him to try to expand that to several other rattlesnake species. So the idea is that our rattlesnakes are carrying, we have a method of attaching them for short-term attachment onto the back of a snake using a surgical bandage, just so the snake carries around this logging device that is logging when it moves and what its temperature is, etc. And then we try to recreate kind of the activity cycles and the lives of the snakes using remote cameras and also direct observation. So that if it works, and it turns out this is tricky with snakes, but if it works, we would be able to do things like tell when a snake is striking, because a snake that is just sitting there motionless and then goes into a strike should have a very unique acceleration signature, like we're showing in this lab strike of a snake, where everything's still and then suddenly a burst of acceleration to strike. It works well with lab-based stuff. It's harder to do this in the field, so we're still kind of struggling with the machine learning part of this. But our hope is the idea that if we record enough snakes doing things like normal lateral undulation, so here's what you might think of as like a walking gait by a snake versus a snake that's kind of running or sprinting away from you, like this snake moving very rapidly through lateral undulation, versus snakes that are just like sitting there in ambush, like one of our prairie rattlesnakes in an ambush, versus snakes that are doing things like trying to ingest a carcass. So here's a speeded up view of one of our prairie rattlesnakes trying to pull in a bird carcass, and we're hoping that if we get enough data, we can resolve the acceleration signatures between that and maybe even other unique snake behaviors. Like for a couple of species, it's really hard to do in the field, but we have got some combat dance behavior, not enough to really be able to resolve this with machine learning models. But the hope is that we can get there and then do the type of thing that others have been able to do with their animals, to kind of take a record of what the snake is doing from these acceleration devices and recreate it to understand details of their natural history, which will bring me back full circle to being like, I think of that technique as just a way for, it's kind of next generation natural history. It's a way for us to study what the animals are doing that supplements our ability to just watch them with binoculars or cameras. The acceleration stuff is part of this next generation natural history toolbox. And I stole that. I should get Tal Levy and his lab credit for that term. They published a paper on this a while ago, pointing out that all sorts of tools that we have, these animal-borne devices and special instruments for observing nature in gory detail and computational advances in community science, it's all just a way to enhance our ability to understand what cryptic and secretive animals are doing in nature when it's hard to observe them directly. So it's expanding our ability to understand these animals like snakes that are really difficult to follow most of the time. Okay, I definitely want to leave time for questions, so I'm going to wrap it up there by saying thank you to all of the students, graduate and undergraduate students over the past many years who have worked on these field projects and lab projects and helped kind of pull together some of the data that I just talked about there. And with that, I will end on a question slide. And Emily, we can go back to you and open it up. They can stop sharing too.
Awesome. Thank you so much, Rulon. That was super awesome. Folks, we have some questions in the chat already. If you have other questions, go ahead and enter those now. We'll have time to get to a few of those. So I'm going to start asking you some of these questions.
Yeah.
Okay, so first of all, one of the questions that we had was whether or not, like, okay, so to what extent does the sidewinder or Mojave rattlesnake, the snake that strikes, kind of know the outcome? In other words, do they show the strike-induced chemosensory behavior only if they have envenomated the prey, or do they kind of try anyway if they're not sure?
No, I think they know if they've successfully envenomated. Like what our video kind of indicates is if it's a really brief contact and they're like kicked off, it's not like they think, oh, I've maybe killed this thing, and they go look for it. They just kind of sit there and then pull themselves together and go hide in a burrow or something. They don't begin that sixth process. But if they do like have a prolonged like contact time where it looks like they inject venom, they almost always like perk up and start like going around, wandering in circles, finding scent trails, and then having this very directed like trail following behavior. So I think they have a pretty good sense of whether or not they've been successful. But that being said, I have seen like northern Pacific rattlesnakes that would envenomate ground squirrels where ground squirrels were resistant to the venom. I know that those squirrels survived because we saw those same squirrels later, but the snake, and because the squirrel was like physiologically resistant to the venom, I don't think the snake had a clue. A couple of times we've watched snakes like go for these really extended searches, wasting a ton of time and energy with what looks like they're trying to follow the scent trail of a squirrel that actually wasn't killed by the venom.
That's really cool.
Yeah.
Poor snakes.
Yeah, I know. I feel for them.
We have a question here about what you know, or even just what you might hypothesize about how python and boa thermovisual acuity compares to pit vipers.
Yeah, so it's thought that it's probably maybe not as good because their membrane system is different. They have like these heat sensitive nerve endings in a membrane, but it's not a suspended membrane like the pit organ. It doesn't have like a cavity behind it that is going to absorb some of the thermal radiance and the interference. So it's thought to be probably not as sensitive, and there are a few experimental studies in the lab that kind of back that up from some, I forget the authors, but there's some people who looked at that, and pit vipers seem to have like be able to detect more subtle contrast or maybe from further distances away. So maybe pit vipers have a slightly better system in terms of like visual acuity or contrast background, but there's so little work done on boas and pythons. I can count on one hand the number of studies where people have really tried to drill into their pit organ work. It doesn't scratch the surface of what we know is present in terms of morphological diversity of the placement and abundance of those different types of pit organs. Really fascinating area. I'm hoping to learn more about it as we get into that.
Awesome, thank you. This question is about the time lapse between the bite and the snake then later finding the prey. Would another animal that finds the bitten prey before the rattlesnake be susceptible to secondary poisoning if it was to ingest that prey?
No, probably not, because the venom, at least for vipers and pit vipers, it's almost all like enzyme based, peptide based. It's like proteins and enzymes that are destroying tissues and digesting stuff. If you swallow it, you just break it down. Like if you like, that's why people who do like sideshow snake stuff back in the day would be like, oh, watch me, I'm gonna like swallow this snake venom and I'll be fine. It doesn't actually hurt you unless it's like injected into your tissues and bloodstream. So if you don't have any like cuts in your mouth or whatever, then you could actually drink snake venom and be fine. So I think any animal, including the snake, that finds an envenomated carcass and eats it, no problem.
And then what if another snake finds it? Will they eat it?
I think so. Like Mark Tashura and I were really interested in that question. Mark did this really interesting kind of lab study on the possibility of kleptoparasitism, which would be like a snake that actually comes across the scent trail of something that was bit by somebody else and finds that carcass and eats it. I still think it could happen, but we didn't find a lot of evidence for it in our captive scenario. It does kind of seem like Dave Shazar back in the day always emphasized, the snake needs to bite something to release this like searching behavior, right? If it doesn't like bite and envenomate something itself, it appears like it doesn't go into this mode of gotta find the carcass, gotta find the carcass, gotta find the carcass. It's like a releasing stimuli. It's like striking and envenomating something. But we also know that snakes scavenge stuff, right? We find them occasionally eating roadkill or something. So to some extent, they're also opportunistic as well as predatory. So I don't think it's impossible for them to kleptoparasitize each other.
All right, there's two more questions. I think one's gonna be quick and then one might be a little bit longer and then we'll end. So the
Okay.
quicker one, maybe, I could be wrong, is when thinking about those signatures from accelerometry. I know you don't have this data yet, but would you
Yeah.
expect that maybe you eventually might be able to tell what they ate based on their signatures? Like, would there be a bulky and low density, like a bird, have a different signature than like catching a rodent?
Yeah, probably not. I don't think, the accelerometry stuff is always going to be limited to like a kind of a coarse resolution of behavioral types. I think I would feel lucky if we just got a signature that worked well in our model that indicated a food bolus passed under the accelerometer. The reason we're attaching them up towards the head is we're hoping this works in the lab, but it's harder with field-based data. We're hoping that that unique kind of pressure that pushes up on the accelerometer when the snake swallows something can be recovered by the AI models to be like, oh, that was a food bolus. Maybe if we had enough kind of validated behavior where we like experimentally feed snakes and like this is a small food bolus and this is a big one, maybe we could even categorize it as like, oh, that was like a relatively small thing versus that was something huge. And maybe like that process of like dragging carcasses around, that might be recoverable. And I think snakes have to do that. It takes them a lot longer to swallow something big than something small. So maybe in the future we would get to some more like fine scale resolution there, but it's not as easy as I'd like.
Okay, all right, so the last question, and sorry if other people have put more questions in there, but we want to try to finish on time, or at least just a couple minutes late. Is a question about natural history and K through 12 science education. This person says that they found that their kids' teachers are emphatically insistent on hypothesis testing for science fair projects.
Yeah.
Argue that my herb-loving kid is doing another valid kind of science when he explores and observes.
Yeah.
Do you think there's a need to reach out to school science teachers about this and other ways of knowing that are sometimes less analytic and more holistic or relational?
Oh my God, this is a great question. I really love it. And it's so funny because I actually have a personal anecdote about this. I got like on 5th place or like last place in my school science project when I was in like 4th or 5th grade. When I can't remember what the details of it, but I had some study where it just like had frogs in a jar in an aquarium and like watched them and something. I thought it was really cool, but it was not, I wasn't like testing a hypothesis. I was like observing animals. So it's really funny that you asking that question because I had like a similar experience. Look, I don't want to like, I'm not going to say that hypothesis testing isn't like the kind of the linchpin of the scientific method. Obviously it is, right? Like once we have enough information to generate like testable hypotheses, that's when we like use the scientific method to test those hypotheses. So it's great and we should be teaching that. But I think what gets lost and what I would say to like a science teacher is like, look, there is a time and a place for hypothesis testing, and then there's also a time and a place for exploratory research where you do not have hypotheses and you want to generate information and quantify it. And that's like, think of it as like a raw material, right? Like us generating like information is providing the natural resource. That's the raw material that gets refined into hypothesis testing. We don't, if you can only test hypotheses if you know something enough about a system to make like a reasonable one. So I think we lose track of the fact that we need a part of our science has to be kind of basic science that is a exploratory in nature, so we can discover things that are unexpected, that we don't have blinders on. I think hypothesis testing, you have to be focused on your hypothesis. You have to put blinders on because you're trying to, like with our snake in the lab, we're trying to exclude any other, like, lurking variables that might be out there that might interfere with the way the snake responds to our thermal stimuli. So we're not actually going to learn anything about that snake other than this one specific hypothesis. Can it detect a pendulum moving in front of a background? I want to know the answer to that question in that particular case, but it doesn't help us. It doesn't generate new material that it could be refined necessarily into other hypotheses. I don't know if that's if it makes a lot of sense, but I think that I would teach the scientific method to grade schoolers as there's two parts to it. There's exploratory observational science that helps us generate raw information, and then we refine that information, that stuff into testable hypotheses, and that's those are complementary. We need to do both things, and so I think it's perfectly valid for a science fair. You have my endorsement for whatever science fair you're involved in, to observe and study animals and then report quantitative information on what you found without going into it with I'm going to discover some specific thing. It's just like, I want to know what a day in the life of this praying mantis looks like or something.
Awesome.
I could go on, but I'll stop now.
Well, it was a really awesome question, an unusual question and a thought-provoking question. And a great answer too. Well, thank you so much, Rulon, and thank you to everyone for joining us today. If some of you arrived late and you missed the presentation, it will be posted to our YouTube channel sometime tonight or tomorrow. And that's our Snake Week YouTube channel. And in addition, I want to remind everyone that coming up actually in 1 hour, so in slightly less than 1 hour at 5 p.m. Pacific time, we have Dr. Rick Shine tonight giving our next Snake Week talk. And then we have 3 more talks this week, and there's a bunch of other stuff happening. So go to snakeweek.org and check out that information. Thank you so much, Rulon, for giving us this presentation today. It's on my favorite thing in the world, rattlesnakes, of course.
And thank you, Emily, for organizing. It was really great, and thanks everyone for coming and listening. This is an amazing event. So huge kudos to you and the other organizers, Emily. We've got to make it happen every year.
That's right.
International Snake Day, Snake Week. Doesn't matter if it's the year of the snake or not. Every year should be the year of the snake.
That's right. That's right. All right. Well, thank you. Nice to see everybody. Take care. See you next time.
Thanks, everybody. We'll see you.